I thank Journal of Cancer Metastasis and Treatment to organize this webinar on how to live a better life of long-term cancer survivors. As invited speaker, I will be delivering this talk, and the title of my talk will be Navigating Long-Term Breast Cancer Survival Using Advanced Cost-Effective Technology in India. Currently, I am Director Research and Development, KIIT University, Bhubaneswar, India. Over last 25 years, we are working in order to understand that tumor microenvironment in breast cancer, especially understanding the tumor heterogeneity, and to develop that diagnostics and targeted therapy in breast cancer. So, breast cancer burden across the globe and in India, among the globe, that highest number of incidents, country like Australia, New Zealand, Western Europe, Northern America, etc. In India, among all cancers, breast cancer is number 1, and according to Globocan data 2020, in India, breast cancer accounted for 13.5% of all cancer cases and 10.6% among the deaths related to cancer. So, therefore, we need that proper strategy in order to increase that breast cancer survival. In this context, Indian Council of Medical Research is going to develop a program how to increase that breast cancer survival, especially stage 2. Currently, that our percentage of breast cancer survival is about 60 to 66% at stage 2 level, as compared to the survival rate in the developed country, it goes up to 90%. So, Indian Council of Medical Research developing a program how to enhance and accelerate the survival rate of breast cancer in India, considering that entire geographical location. As I said, that understanding tumor microenvironment is very important. Not only targeting tumor cells, there are many stromal cells which include cancer associated fibroblast, many immune cells. They express varieties of genes and proteins, and in a coordinated way they control the growth of the tumor at primary site, ECM deposition, angiogenesis, and metastasis at distant site. So, as all of you know, breast cancer, they have 4 major subtypes: luminal A, which is ERPR positive and HER2 negative; luminal B, where all 3 positive, HER2 overexpressing; and triple negative or basal like breast cancer, where ERPR and HER2 all 3 negative. Country like India, we found that majority of the breast cancer patients, they are developing breast cancer even at the age of 27, 29, 31, and they are belongs to triple negative breast cancer, very aggressive, and they are developing metastasis, not only that lymphatic metastasis, but other metastatic area like lung, liver, brain, and bone. There is another subtype of breast cancer called IBC, that is inflammatory breast cancer, where that entire organ become inflamed. So, it starts with that little lump here and there at the benign stage, then the tumor grow at the primary site, then first target to that lymph node. Once it target to the lymph node, then it's metastasized to other organs such as lung, liver, brain, and bone, as I mentioned. So, in this connection, we developed a multicentric national program through Department of Biotechnology, Government of India, that is called DBT VNCI program, in association and collaboration with Tata Memorial Hospital, KIIT University, ACTREC, Institute of Bioinformatics, and National Institute of Biomedical Genetics, India. The objective of this program to understand the molecular mechanism in hormone refractory breast cancer. So, key objectives: to establish the primary culture and cell line from hormone resistance and sensitive breast cancer patients' tissue, to develop that patient derived xenograft PDX model, to screen the new drugs, testing efficacy of selective known drugs, and to identify the therapeutically relevant biomarkers in patient derived xenograft models. And finally, to identify and validate the novel therapeutic targets from breast cancer patients, patient derived xenograft models, and primary culture using phosphoproteomics and genomics approach. So, we are the first group laboratory from India established that patient derived xenograft model, PDX model, using these hormone resistant breast cancer patients starting from generation 1 up to generation 5, and these are the representative tumor, and we have characterized these G1 to G4 generation and compare with the patient's data, and they are showing that same hormone positive breast cancer by analyzing the specific hormone positive signatures like the status of expression of ER alpha, PR alpha, they are showing positive, along with EMT specific signatures like N-cadherin, bimentin, proliferation marker like Ki67, they are showing positive, along with pan-cytokeratin. So, this paper recently published in Oncology Reports in a collaborative way. Next part of this talk, I will be talking with a protein, chemokine like ECM associated protein called osteopontin. It interacts with its receptor integrins, especially alpha B beta 3 integrin and CD44. Once this chemokine like protein interacts with the receptor, it regulates cell-cell communication, cell matrix interaction, ECM deposition, angiogenesis, tumor growth, and metastasis. So, over last 25 years, we actively shown that both osteopontin and its receptor, they overexpress most of the cancer tissues and cell lines, including breast cancer. So, therefore, osteopontin act as early diagnostic markers and therapeutic target in breast and other cancer. So, this what we have recently shown that like many other growth factors or chemokine, which plays important role to differentiate fibroblast to activated fibroblast or myofibroblast, we ask this question whether osteopontin being a chemokine, whether that differentiates or transdifferentiates resident fibroblast to myofibroblast or cancer associated fibroblast. In this context, one of my students, Dr. Ramesh Butti, who is currently University Texas Southwestern Medical Center, Texas, US, used during his PhD at my laboratory at National Center for Cell Science, Pune, and thereafter at School of Biotechnology, KIIT University, demonstrated that when used that human fibroblast cells like BJ cells exposed with osteopontin, found that the normal fibroblast differentiates into myofibroblast or cancer associated fibroblast, thereby the expression of CAP or cancer associated fibroblast specific genes or proteins like alpha SMA, PDGF receptor beta, FSP, FAP alpha, their expression goes up, which he has shown both at protein level as well as RNA level. Then we use that parental TNBC cells MDMB 231 and knock down osteopontin with a lentiviral virus system in parental MDMB 231 cells. Independently, either parental MDMB 231 cells exposed with normal fibroblast or osteopontin knockdown 231 cells exposed with fibroblast, and then check the expression of these CAP specific signatures and marker. We find when we knock down osteopontin in parental breast cancer cells in association with fibroblast, the expression of CAP specific signatures like alpha SMA, FSP FAP alpha goes down as compared to parental breast cancer cells in association with fibroblasts. This suggests that tumor derived osteopontin reprograms fibroblast to myofibroblast. Then we check that what is the status of transcription factor twist1, and we found that both twist1 as well as trauma derived factor SDF1, their expression goes up when we use that parental breast cancer cells MDMB 231 in association with normal fibroblast, but when we knock down osteopontin, then that expression and activation of twist1 as well as SDF1 goes down. This suggests that tumor derived osteopontin activates twist1 transcription factor through transdifferentiation of fibroblast to myofibroblast through MMT, mesenchymal to mesenchymal transition, thereby that expression of stroma derived factor 1 SDF1 and alpha smooth muscle actin alpha SMA goes up. Then we proved our hypothesis by developing two preclinical animal model, one carcinogen induced mammary tumor model in osteopontin wild type and knockout mice, and we found that osteopontin knockout mice, the size and growth of the tumor is much lesser than wild type mice derived tumor. I am not going to show that data. Second model where we co-implanted that MDMB 231 cells along with fibroblast and co-implanted to that immune deficient Nodskid mice. Parallelly, we knocked down osteopontin in MDMB 231 cells and co-implanted with that fibroblast. So, I am showing this data in this next slide. So, when we co-implanted that MDMB 231 along with a fibroblast, the size and growth of the tumor much higher as compared to osteopontin knockdown 231 cells co-implanted with the fibroblast. We analyze these tumors and by immunohistochemistry and protein and RNA level, but I am not showing those data because of lack of time. What I am going to show you that using that breast cancer patients starting from grade 1, grade 2 and grade 3, we find in the higher grade of breast cancer patient tissue, the level of expression of alpha SMA and osteopontin are high as compared to lower grades. Most importantly, alpha SMA expression is very high in grade 3 breast cancer patients, and alpha SMA positive breast cancer grade 3 breast cancer patient tissue, we find that twist1 expression also high and they co-localize along with alpha SMA positive breast cancer. This suggests that alpha SMA positive breast cancer, that means the alpha SMA positive cancer associated fibroblast, plays very important role for the growth of the tumor in response to osteopontin in breast cancer patients. We have also done the correlation studies where we have shown that there is a very good correlation between the expression of osteopontin along with a CAF specific signature like a fibroblast associated protein FAP, then alpha SMA, then twist1, etc. And using that tumor microarray or tissue microarray, tumor tissue microarray, we have shown that both the expression of alpha SMA and osteopontin are very high in that patient tissue as compared to normal healthy individual. So, we propose that osteopontin, irrespective of interacting with integrins or CD44, it activates the twist1 through PIK3K and ARC pathway, and this twist1 ultimately controls stroma derived factor 1 SDF1, FAP as well as that other CAF specific genes and proteins, and these SDF1 and alpha SMA, they ultimately controls that tumor growth in breast cancer. And this paper recently published in Oncogene 2021. So, we have earlier also proven that osteopontin basically regulates HIP1 alpha dependent VEGF expression and tumor angiogenesis in breast cancer model. By analyzing that breast cancer patient tissue starting from fibroadenoma to ductal carcinoma in situ and infiltrating ductal carcinoma, we find that especially in infiltrating ductal carcinoma, that expression of osteopontin and HIP1 alpha is very high as compared to fibroadenoma. This also suggests that osteopontin plays very important role in the growth of that tumor at the aggressive tumor in breast cancer patients. So, considering all these findings, we proposed and supported by many other researchers that osteopontin act as a therapeutic target in breast as well as other cancers. We use aptamer based technology, siRNA, shRNA, CRISPR-I technology, CRISPR-Cas9 technology, as well as by developing the druggable small molecules which can disrupt the interaction between osteopontin and integrin or CD44 and thereby regulates or controls or suppress or regress the tumor growth in breast cancer as well as other cancers. Now, I will be showing how that osteopontin act as a early diagnostic marker. So, Professor Ann Chamber from Canada has shown that serum plasma osteopontin level have prognostic value in metastatic breast cancer, and they have shown by ELISA in normal healthy individual the level of osteopontin is about 123 nanogram per ml, but patients in metastatic breast cancer it goes 138 to 1312 nanogram per ml. Considering that, we in collaboration with Ruby Hall Clinic and Jahangir Hospital, Grant Medical Foundation, Pune, studied that osteopontin and COX-2 cyclooxygenase 2 can act as a potential marker in breast cancer, where in the normal peripheral tissues by immunohistochemistry we do not find any osteopontin and COX-2 expression, but when we use the HER2 positive breast cancer patient's tissue and by immunohistochemistry we find the level of osteopontin and COX-2 level are very high. That suggests that osteopontin and COX-2 can act as a potential marker in HER2 overexpressing breast cancer. Similarly, most importantly in triple negative breast cancer where ER, PR and HER2 are negative or absent, there also we find that both osteopontin and COX-2 expression are very high. So, this also suggests that osteopontin and cyclooxygenase 2 act as a potential marker for detection of cancer in TNBC or triple negative breast cancer. So, these studies we have done with 269 patients with about 41 patients with HER2 overexpressing and 228 patients with non-HER2 overexpressing with grade 1, grade 2 and grade 3. These data also published earlier. Now, we are moving further in terms of diagnostics related to early detection of breast cancer in collaboration with Professor George Weaver from University of Cincinnati, Ohio, US, where we are developing a PCR based blood test for detection of breast cancer recurrence using various splice variants of osteopontin. This study is going on. Recently, we filed patent on this. Targeting osteopontin or osteopontin as a early detection system or early diagnostic marker as well as a therapeutic target. In this connection, we publish multiple papers in Cancer Research, Oncogene and other journals, and some of the report published in Times of India earlier days. With that, I have about 23 students completed PhD degree with me, majority of them they are in United States. Currently, 7 PhD students working at School of Biotechnology, KIT University, postdoctoral fellow and research associate, research officers, project JRF and many collaborators including Professor Lucio Meli, who was that founder editor in chief of JCMT, and he is a professor from Louisiana State University Health School of Medicine, New Orleans, along with many other collaborators from IIT Bombay, Saroj Gupta Cancer Center, Curtin University, National University Singapore, similarly Hanover Medical Center, University Oslo, Murdoch University, Perth, Australia, and then many funding, national and international funding from Department of Biotechnology, Department of Science and Technology, Indian Council of Medical Research, Indo-German funding, and many students who work under this project under different capacity at National Center for Cell Science, Pune, along with School of Biotechnology, KIIT University. With that, I like to thank Professor Achyuta Samant, founder of KIIT University, and Professor Sasmita Samant, Vice Chancellor of KIIT University, for their continuous support to further progress, active progress of that research and development department of KIIT University, Bhubaneswar, India. Thank you very much.